I will figure that out as we go. Well, welcome everybody. Good to see you all. Can you guys hear me okay? Give me a thumbs up or nod. Okay, good to see you. Thank you so much. Appreciate you guys. And Dr. Dixon, are you on?
I'm here.
All right, let me make you co-host.
You had to become an IT professional here, Dondee.
Apparently. Yeah, that wasn't in my job description either. I just made you co-host. Did you get it? Are you co-host now?
It looks like it. I can share, it looks like. So
awesome.
Ready to rock and roll.
All right. Well, welcome everybody to another Points on Product Zoom call. I am Dr. Dondee Nettles, apparently not filling in very well for Rye Fry or Patty and Greg, but please give me some mercy since I'm working on their channel. In fact, down if you look at the bottom left corner, that is not my name. That is the name I am assuming today, and I'm not apparently doing a very good job. But welcome everybody. We're super excited because we're really going to go back to the beginning of what makes LifeVantage so special, why are we so unique, and what really facilitated that process? Well, it's really all about the studies. If you've been involved in LifeVantage very long, you've heard about the studies, you've heard about the scientific validation, where we're literally going to go back chronologically to the study that started that conversation, to the study that put this fledgling network marketing company that literally no one had ever heard of on a global map by doing something that pharmaceutical companies do. Big name pharmaceutical companies and other medical universities and other prestigious companies do, and that is have third-party initiated funded studies. Now, the one we're going to go over today, that Dr. Brian's going to go over today, was we would consider it the pilot study of really what got LifeVantage on the map, but it was incredible. It was profound. The results were profound. It was a human study. Forget the mouse studies, forget the human culture studies. We went right into a human study to begin with, and it's paid profound dividends. But this is the study that really got it all started. And before I talk about that, I want to share with you who's going to be exploring this with you, who's going to be sharing this with you. And it's none other than our illustrious leader, Dr. Brian Dixon, our Senior VP of Product Development and Senior VP of Research and Development here at LifeVantage. If you're not familiar with Dr. Dixon, I don't know what planet you've been on. You should be. Everybody is. But let me I'm going to give you a cursory version of his bio. Dr. Dixon earned his PhD in molecular and cellular biology from Oregon State University in affiliation with the Linus Pauling Institute, and his research focused on underlying biomechanical or biochemical mechanisms of the aging process. He has authored several scientific peer-reviewed manuscripts on topics such as antioxidants, cellular signaling, gene regulation, stress coping mechanisms, inflammation, immunity, the aging process itself, Nrf2 activation, and research on nutrients such as glutathione, vitamin D, plant polyphenols, and other phytonutrients. So I gave you a brief, abbreviated version of Dr. Dixon's background because what he's going to really get into is the first study that LifeVantage did to talk about this whole thing about Protandim. And we didn't call it Protandim Nrf2 Synergizer back then. It was just called Protandim because there wasn't any other Protandim products. That was much later. But without further ado, and I know we're already behind the schedule, Dr. Dixon, please take it over and share with us your incredible knowledge on this study and help us understand the relevance and the importance of what it really says to us.
Yeah, thanks, Dr. Nettles. And man, I can tell you were in radio at one point in your career. You got a great voice for it too. So I appreciate the kind words, the introduction. I think you set it up beautifully, so I appreciate that. But yeah, kind of like you alluded to, we're going to go right back to the basics. And I would argue that if you're only going to read one of the 31 publications around Protandim, this should be the one. Forgive me if I hope it was circulated, but just in case it wasn't, here is that article. And so the title, the induction of human superoxide dismutase and catalase in vivo. So that means actually in real living people, a fundamentally new approach to antioxidant therapy. So if you remember from last week, and if you didn't get a chance to watch that video, maybe go back and watch that. I think Dr. Nettles and myself, we did a great job of kind of laying out how we approach scientific manuscripts. but we'll go through a lot of that again today. And we're going to go in depth. And I figure since we're going through scientific studies, I have the right to show some science for real. So I'd like to get a little bit deep into a couple of things that I think are important just for background information. Again, I'm not trying to make you all little scientists in your own right, but I think just having a basic understanding can go a long way, if nothing else, just with kind of your stature and your confidence with how you might be approaching people. So let's kind of jump in. And again, just to kind of really high level how we approach a scientific manuscript, usually I just go right to the title. Usually the title is that kind of conclusion, and it'll have some key words in it that'll help make it easy to find in a search. So like we talked about, this one's all centered around utilizing Protandim in people and then measuring superoxide dismutase and catalase, and then some actual clinical markers that are resulting from the findings of this study. Like I mentioned, I do like to go and look at the journal that it's in. Free Radicals of Biology, or Free Radical Biology and Medicine, is a fantastic journal. I looked up the impact factor. Again, something we talked about last week. The impact factor of this journal is 7.376. So it's actually, that's an amazing impact factor. This is a really well-respected scientific journal. And then I like to see kind of, the flow of when the publication was first submitted, when it was actually then accepted for proposal or for publication. You can see here, there's some interesting nuance here. So it was first received in June of 2005. And then what this revised in 24th of August means is that the reviewers came back to the authors and said, oh, we'd like you to do a couple of things, make a few little changes. So what's interesting is this is actually a pretty quick turnaround. So from June to August, it shows either it was great research or the research was pretty buttoned up and they only had small revisions to make. Sometimes you'll see a massive, even couple year lead time between some of these dates, and that'll show that maybe the publication wasn't that well buttoned up when it was first submitted and the authors had a lot more work to do, and then pretty quickly published 4 days after it received those revisions. I also mentioned I like to look at the author list just to see if it's anybody I know personally or an expert in the field. So as we kind of come across here, there might be a gentleman's name that you recognize, Joe McCord, the original inventor of Protandim as we know it today. And then from there, I just kind of start picking my way through the different sections. So we'll have that abstract, the introduction, the methods, the results, the discussion. Sometimes the discussion and conclusion are wrapped into one. And then there's, of course, all the reference list down below. I also do like to look at the research institutions as well. So you can see this is mostly from Colorado, University of Colorado. And I know where Joe McCord was home based for quite some time. But then as we come down, we read the abstract, and the abstract is really a great outline of what you can expect inside of the publication. And so the abstract can give you some pretty good insight into what you're going to be reading further on. But I also caution you not to just take all of the findings only from the abstract, because usually when the abstract's written, you're confined to a certain number of words, usually just a small couple hundred different words in the abstract. So it can be pretty truncated, and you could miss some pretty interesting details, and some of those were actually missed in the abstract that we're going to go through a little bit later. So again, this abstract serving a lot as an outline, not a whole lot of introduction here, but they just jump right into the methods of what they did in this human clinical study. And then they get down into really what they found. So utilizing an assay that I think many of us are familiar with, called T-bars, they were able to show a strong age-dependent increase, right, in their T-bars when people were unsupplemented, right? And then we'll also see that there was an age component to that as well. But then after 30 days of supplementation, the T-bars declined by an average of 40%. So has anyone heard the claim that we make that you can reduce your oxidative stress by 40% in 30 days? That claim exactly came from this publication. And because they did such a good job experimentally of laying that out there, we're able to make that claim in a compliant manner. And it's just an absolutely wonderful claim, and not one that I think anybody else in the industry can even come close to saying. We get into the introduction, and remember, the introduction is really giving a little bit of background information and how this new research fits into what is already known. I do like reading the introductions because it can give you some good insight and some good scientific background. For example, something I've pulled out of this paper is this: oxidative stress is now recognized to be associated with more than 200 different diseases, or we can compliantly say health concerns. Okay, so I just modified disease to health concerns. So what a great little fact to throw up. And I think Dr. Nettles and I were presenting together, was it last week or the week before? Gosh, it all blends together. But right, I had a slide that says, why is oxidative stress important? And that's because it's recognized to be associated or even underlying over 200 different health concerns. So you can get some good background information, maybe some good fodder for presentations you might be giving. But anyway, it just does a great job of setting the stage for what's going on. The materials and the methods, remember, I don't necessarily spend a lot of time on this up front, but I know that it's there in case I need to go back to it to maybe get a little more detail into an experiment or a result that they may have found. I do. Well, we'll come back to that in just a second. And then we get down right into the results. And this is where this paper, I think it's really, really fun. Remember, I like to read kind of these headlines. So effect on lipid peroxidation and T-bars. Okay, so I think there's an interesting paragraph here that I'm going to read, and that is: the primary objective of Protandim supplementation is to decrease oxidative stress. Our endpoint to assess oxidative stress in this study was T-bars, which measures a family of lipid peroxidation products, mostly lipid hydroperoxide. So a lot of chemistry and science jargon going on here. But these things break down during analysis to yield another compound called Malinda, which reacts with this T-bar substance that's called 2-thiobarbiteric acid. I did okay on that one, to yield a chromophore, which means something that's visible by light that can be measured at a specific wavelength. So some pretty fun background going on there. So utilizing those T-bar techniques, we come into figure 1. And this is where there's some really cool nuance that you would never pick up in that if you only read the abstract. So we're going to spend a little bit of time, and we're going to talk about this T-bars assay, and we're going to talk about some of the results, and some of the results they call out outright, but then also some, I think, pretty important findings that really aren't dwelled upon. And maybe it's with the luxury of hindsight and the future research that had come out after this study that we can go back and tease this out. But let's spend just a minute looking at T-bars, if you will indulge me. We're going to get into a little bit of chemistry.
While you're looking that up, Dr. Brian, with that jacket you're wearing, makes me think of Inspector Gadget. I keep thinking these arms are going to come out and go do something. I'm loving it.
That's too funny. It's just so cold always here in the office. All right, let me share this. Bada bing, bada boom. All right, I hope you guys are ready for a little bit of chemistry. Again, you don't need to know this, but I love molecular biology. I love chemistry because it explains what's going on. Nothing is magic that's happening inside of the cell, whether it's molecular biology and following different molecules around the cell, whether it's activating genes in our DNA, or whether it's oxidation of lipids. It's all chemistry, and it's all very predictable, if you will. So remember, the T-bars assay was measuring something called malindialdehyde, and that's this compound down here at the bottom left. But where does malindialdehyde come from, and why is it important to be measured? The place where malindialdehyde gets its start is from these fatty acids we call PUFAs, or polyunsaturated fatty acids. Now, this has always baffled my mind. Nobody knows the real answer to this, but we all know that PUFAs, polyunsaturated fatty acids, like omega-3 fatty acids, are incredibly beneficial to our health. Yet it's only PUFAs that are able to be oxidized, and it's because of this chemical structure with them having double bonds. And if you remember the presentation we did on omega plus and fatty acids, I can tell here's carbon 1, 2, 3. So this is actually an omega-3 fatty acids. So what will happen is, because of the way these double bonds are laid out, it actually makes it really easy for a free radical to attack a location between these double bonds. So it can only be here and here. So it's because of the double bonds why these polyunsaturated fatty acids are so susceptible to oxidative damage. So once they gain that extra electron, you'll see some shuffling of those electrons that goes on inside of that fatty acid tail there. Then what happens is something called a peroxide, right? Like hydrogen peroxide, even superoxide is a peroxide, will come and attack this compromised site and will actually adduct it, adding these two new oxygen molecules there. That's also a free radical, and why it's called the lipid peroxyl radical, because you have these peroxides that come in, attack, and now it's a free radical. And these things get incredibly potent and can be incredibly damaging to structures inside of the cell. But what we're trying to ultimately get to is malindialdehyde. So you get some of these electrons shifting around again, forming this little ring, but ultimately cleaves again at that site where that free electron is dancing around. And we end up with this compound called malindialdehyde. So T-bars, remember, is measuring malindialdehyde. Now, why do we measure malindialdehyde versus just the actual free radicals or the free radical species? And that's because these free radical species tend to be incredibly short-lived because they are so reactive. So to be able to look at them in a steady state manner becomes incredibly difficult. So we look to measure more stable endpoints that can be a marker of that free radical or oxidative damage that's occurring. Dondee, I don't know, how was that? Was that clear as mud? And do I have everybody just regretting joining the call?
It was clear and somewhat understandable, but I think it's something great for the person who doesn't really understand anything you're talking about to understand that there is a massive complexity to this, that to the people that are scrutinizing it and looking at it, it's going to be a big deal. To your average person, it is probably all Greek, what you just mentioned, but for somebody who understands that we are different, to hear you go and explain that, it really just sets it apart that, you know what, I don't really know what he's talking about, but it's a really big deal. And the people that do know what he's talking about are going to see that. I really think that's what our science has always given us, is this edge that people that don't understand it, that's okay. But the people who do truly get it, and it sets our company apart instantly. So I think you probably, you really did a great job in simplifying it for most people who've never taken chemistry or this level of chemistry, it might have gone right over their head. But suffice to say, if that was you and it went over your head, there are people that will look at this someday as a result of you introducing it to them. They're going to go, oh my gosh, I've never seen anything like that before. That's the power of what we really have here.
Yeah, I appreciate that, Dr. Nettles. Thanks. The take-home message, the only thing I want you to take away from this is that molecule, malindialdehyde, is a marker for oxidative stress and oxidative damage that's occurring inside of the cell. But let's actually get down to the assay and what's going on, okay? So remember, here's that malindialdehyde molecule. As we're experiencing oxidative stress inside of our cells, tissues, and organs, malindialdehyde is actually a really reactive and quite a nasty compound. I mean, it's malindialdehyde. So like we've all heard of different aldehydes like formaldehyde. I mean, that's what we basically pickle people with after they die. And the reason is because they're so reactive. It just sticks to everything and basically links everything together. And I don't know, whatever gets pickled just turns into one solid mass, basically. Anyway, so this is an incredibly reactive compound and our bodies don't want it around, so it excretes it, and why it's probably ultimately ending up in the bloodstream. But what we can do is take blood, put it into our test tube, our T-bars experiment, incubate it with the actual reactive compound, that thiobarbituric acid. So that's why we call it T-bars and don't call it by name. And then again, you get chemistry happening. So you'll have the first T-bars reagent react with that malindialdehyde forming here. And then it turns out a second molecule of that T-bars assay reacts and you form this really stable end product. And if you remember that 432 nanometers, we can shine light at this new molecule that we formed as a result of that chemical reaction, and it will specifically emit light at 532 nanometers. Okay, so we can then specifically look for that range of wavelength of light. If there's none of this formed, we won't see any of the light and it will be a low oxidative stress. Whereas if we see a lot of this T-bars compound, there will be a lot of oxidative stress and oxidative damage. So I hope that helps give just a little bit of background of the chemistry that's going on for this assay, because you're going to see T-bars a lot during this publication. All right, let's go back down to figure 1, and we'll talk about some of the, I think, really interesting findings. So here in figure 1A, what they are looking at is really all of the subjects that were in the study. Now, what's interesting to point out here is, can you see how some individuals had a really low oxidative stress and other individuals had a really high oxidative stress? And I should say this is plotted as a function of age and that T-bars number on the y-axis. So really, as a population, we are all over the place. Some individuals already have a low inherent oxidative stress. Some have a really high oxidative stress. And one question I get asked quite often is, why do some people feel a difference relatively quickly taking Protandim Nrf2 Synergizer, and others maybe don't feel anything? And part of that explanation is probably due to where they might be falling in terms of their oxidative stress. If they're low, they're probably relatively healthy. And by definition, it's really hard to make a healthy individual even healthier. Whereas individuals with sky-high oxidative stress, when we're able to bring that down, that's when they're feeling the direct benefits of Protandim Nrf2 Synergizer. Okay, but keep this in mind, this massive distribution. Then what they did in figure 1B was they looked at the differences between males and females. And statistically speaking, they saw no difference. So it doesn't matter if you're a man or a woman, you have the same likelihood to be experiencing oxidative stress. here in C, they broke out the subjects into two different groups. And those were individuals that were already supplementing with dietary vitamin antioxidants like vitamin C and vitamin E. And then the other group were those that were not supplementing with C or E. And interesting, and we'll get to it in just a minute, but one thing they point out is that individuals that were taking the highest dosages of vitamin C and vitamin E actually had higher T-bars levels than those that weren't taking anything. But this is one of those nuances that I think is worth pointing out. So basically D comes from panel A, okay? But you see how this massive distribution has gotten incredibly tight, and every single individual in the study experienced a reduction in their oxidative stress levels as measured by T-bars. So not only notice how it was lowered, but also how tight this distribution got. So basically everyone has the potential to be able to lower their oxidative stress and get down to a healthier spot. Dave, you're making me hungry. I'm starving. All right, if we go down to figure 2. If we go down to figure 2 here, this is a figure I love to show during presentations, okay? The reason for that is what they're looking at is the amount of superoxide dismutase and the amount of catalase. So these two antioxidant enzymes that Protandim is increasing. And they looked at this for up to 120 days. What we see here is an additive effect for 4 months of taking Protandim. So we can measure this increase beginning in 30 days of taking Protandim that continues on to 120 days. So there is this massive additive effect that comes by taking Protandim long-term. Again, another question that I get quite often is, should I only take it for a month so that I experience that 40% reduction? Well, this clearly says no. The longer you're on the product, the higher these antioxidant enzymes become in the blood cells that they measured. Then we move on to figure 3, right? Another one of my favorite figures to discuss. And again, we'll see some nuance here. So here would be an example where I would want to go back to the methods, because I hadn't seen anything breaking up different groups. But the fact that they're telling me there's a group 2 here means that there must have been a group 1 as well. So if we cycle back up to the methods, we can get a better understanding of what the difference between group 1 and group 2 is. And so we'll kind of scan this a little bit and we'll look to see where they start talking about the different groups. And it turns out here I see group 2, so somewhere up above must be group 1, sure enough. So here's an example. We can go back to the method and get some understanding of what it was. So group 1 consisted of 20 subjects that received the full supplemental dose at 675 milligrams, right, in a single capsule. Whereas group 2 consisted of a pretty small group of 4 individuals that were only taking a half of a dose, okay? So 338 milligrams. Okay, so now we know the difference between group 1 and group 2. That will help us better understand figure 3 here. So here in group 2, these subjects that were supplemented with basically a half a dose of Protandim, look what happens to their plasma T-bars levels over time. And here's some of that nuance. So what we can see is that Protandim is working within 5 days. Okay, so within 5 days of these study subjects taking Protandim, you could see this decrease in their T-bars levels. Okay, so within 5 days, their oxidative stress levels are already coming down, and that trend continues for up to 30 days. And remember, this is only with half of a dose. What's so interesting here, and probably because it's such a small group, is they actually saw a 50% reduction in their oxidative stress levels. So maybe we should be saying that Protandim can lower your oxidative stress by 50% in just 30 days. But here is another one of those nuances. This dark bar right here, or dark point, is actually the group 1 subjects that were taking the full dose of Protandim. So you might be saying to yourself, well, it might just be worth breaking the capsule in half and I'll get the benefit. But what this dark bar is showing is, indeed, it is better to take the full dose of Protandim because we see an even lower level of oxidative stress. Now, in biology, I particularly like to see studies like this where they vary the dose, and it's what we call a dose-dependent response. So if an activity, a biological activity, is real by, in this case, a supplemental intervention, you should see a little bit of an effect at a low dose, more of an effect at a higher dose, and then hopefully the largest effect at the highest dose. And this is exactly what we see here. And the reason that's important to me is it's alluding to the fact that there's a real biological mechanism that's going on in how whatever intervention might be working. And in this case, it was Protandim. So like I said, this is a relatively short paper, just 3 figures in the study, but an incredibly powerful study. And again, something that we've based a number of our claims on. But where I think this paper also gets really, really intriguing is with the discussion. So I'd like to spend maybe a few minutes kind of going through, I think, some highlights and some I think really cool sound bites that can come out of the discussion. And again, if you're not familiar with the discussion, remember the introduction is talking about what's already known in the scientific literature, and then what this new research is going to do to help expand that. The discussion takes the results that were found and puts it into the context of that larger scientific literature. So a couple of things I want to point out going through the discussion. I think this first sentence is incredibly important. And remember, this study was done back in 2005, 2006, okay? So Protandim was pretty early on at that point. So no toxicity or evidence of other unwanted pharmacological effects of Protandim were noted at either level of supplementation. So either the low dose or the high dose. Some other interesting things I want to point out. I think these couple of sentences are pretty powerful. The T-bars test is the most widely used in the literature to assess lipid peroxidation and was selected because it allows direct comparisons with the largest number of studies from other laboratories. So what they're saying there is that this T-bar test is largely and widely accepted, especially by laboratories that are well-versed in studying oxidative stress. In particular, a recent study found plasma T-bars to be a predictor of cardiovascular events in patients with established heart disease, independent of traditional risk factors for inflammatory markers. So what they're saying here is that traditional markers like high cholesterol, high blood pressure, weren't as good of a predictor of having a cardiovascular event as your measure of oxidative stress in the blood. So when we ask or get asked, why is oxidative so important and why does it matter? It's because it's underlying so many diverse health conditions. And then they go in to talk about the T-bars assay, the T-bar assay itself, and some of the potential weaknesses that are associated with the T-bars test. And it might be fair criticism, it might not be, but the fact of the matter is that every single experimental assay that's out there that measures free radicals, oxidants, oxidative stress. They all have their pros and their cons. And so we kind of need to know what those are going in. So they talk about some of the criticisms, but then if you remember, there's always the but statement that happens, right? These are the potential drawbacks of the assay, but we believe it to be very valid because they say it is the ability of T-bars to test collectively, right? That lipid peroxidation, think lipid damage by oxidative stress, including the precursors to break down malindialdehyde. And basically what they say here is that we, the authors, and other experts in the field regard this as a really strong assay to be able to measure the consequences of oxidative stress. I love these couple of sentences right here. So remember, they were measuring your oxidative stress in the blood, but specifically they were looking at erythrocytes, which is the fancy way to say red blood cells. So erythrocytes or red blood cells have a circulating lifespan of 120 days. Now, isn't that intriguing why they looked at 120 days and the levels of SOD and catalase? So therefore, by the end of our experimental study, we would expect that 100% of all those new red blood cells have, or those old blood cells have turned over, new ones put in their place, reflecting a new steady state level of both SOD and catalase induced by Protandim. So what that sentence is really saying is that dependent on how quickly a tissue is turning over, and in this case, red blood cells would kind of be a fast medium type of turnover for our cells. But the slower that a cell turns over, the more likely you are to benefit from taking Protandim for an extended period of time. So some of the tissues that turn over the most slowly in our bodies would be our heart cells, our brain cells, nerve cells. So you can imagine if we extrapolate this data just a little bit, those longer lived tissues and cells are going to experience a better benefit by taking Protandim for even longer. This paragraph is also, I think, an intriguing paragraph. Remarkably, this age-dependent increase in T-bars, and they're talking about the back to figure 1, excuse me. But this age-dependent increase in T-bars, so remember that figure 1A, was almost completely abolished, meaning everybody basically returned back down to a baseline level of oxidative stress with an overall average reduction of that oxidative stress marker by 40%. And again, where that claim is coming from. I think this is another great couple of sentences, but there was no evidence that the subjects showing the lowest initial levels of oxidative stress were in any way compromised. Okay, so that's an interesting statement. Why would individuals who already have the lowest oxidative stress potentially be harmed by taking Protandim? And again, why that safety data is so important. Okay, so what are they talking about? The worry was initially, right? Remember, this is incredibly innovative in the space at the time. We love to talk about oxidative stress, but believe it or not, and to make everyone's life as complicated as possible, there is also something called a reductive stress, right? It's the opposite of an oxidative stress, but can also be as potentially deleterious as an oxidative stress. So what they were thinking about in this case was, okay, if these individuals already have the lowest levels of oxidative stress, can Protandim actually take them too low and drive them into a reductive stress? And the answer is absolutely not, right? Compared to modest elevations in SOD and catalase were achieved, an outcome considered remote and therefore possible due to the recognitions that problems can result from too much SOD as well as from too little. So what they're talking about is the balance between oxidative stress, a nice happy medium, and then a reductive stress. The results from this study show that the subjects benefited to varying degrees from the protandim-induced elevations of SOD and catalase activities. Even those with the lowest initial levels of lipid damage or peroxidation, oxidation, however you want to say it, saw modest declines, and in only one subject was the post-protandim value slightly higher than the initial T-bars value. So again, some nuance in there is that if somebody's already experiencing a low level of oxidative stress in their body, Protandim isn't going to take them down into any sort of danger zone. But yet, even those individuals that were at the lowest levels still experienced a slight decline in that oxidative stress. And the main reason why Protandim wouldn't be driving those low individuals down into a dangerous reductive stress state is because, remember, SOD and catalase and these other hundreds of enzymes that we know we can upregulate or activate with Protandim are also self-regulating. So they can basically sense the oxidative stress environment around them. When it's high, they keep working. But as that oxidative stress starts to come back down, those enzymes can also sense that and it shuts themselves off. So another benefit of activating our own antioxidant enzymes is that regulatory component, right? That self-regulatory component. And why are we fighting biology? Our bodies have already sorted this out. Let's just help our bodies help themselves through this activation process. Here is another, I think, really interesting couple of sentences. Okay, so basically what these couple of sentences is saying is you end up in a vicious downward spiral. So once your oxidative stress starts to increase, okay, so let's say there's a lot of superoxide around, for example, this first sentence, superoxide can actually inactivate both catalase and glutathione peroxidase. Okay, so these are two incredibly important antioxidant enzymes, but yet free radicals and oxidants actually will inhibit these antioxidant enzymes, which means that your oxidative stress is going to go even higher. You're going to get more inhibition of those antioxidant enzymes, and your oxidative stress is just going to continue to spiral out of control. So thus, in a system experiencing substantial oxidative stress, the entire group of antioxidant enzymes may be subject to partial inactivation by those unscavenged or unneutralized oxidants and other free radicals, right? So we want to stop that vicious cycle from occurring. Let's get our oxidative stress down. And in fact, that's going to also help SOD and catalase work even better. And then just really three more paragraphs I want to read to you, if that's all right. But I think these are also just great to go through. So the first here, the possible effects of upregulation of antioxidant enzymes on longevity and lifespan should not be unmentioned. So-called free radical theory of aging, right? Something we talk about all the time, first proposed by Denham Harmon in 1956, may not account for all aspects of aging, but is nonetheless widely held. Okay, so what are they saying there? They're saying really this is really one of the oldest and most appreciated theories of aging. But then they go on to talk about the mitochondria. Gosh, don't we have a product for that as well? So mitochondrial metabolism is known to be a major source of superoxide generation, as well as non-free radical oxidant hydrogen peroxide. So the mitochondria are producing a double-edged sword in the production of oxidants, but also those peroxides, if you remember, that can damage the lipid molecule to ultimately make that malindialdehyde. molecule. So here, the maximum lifespan of Drosophila melanogaster, these are the fruit flies that we have flying around our fruit bowls in the middle of summer. It's actually a really well-accepted experimental model. Also easy to do genetic studies. So what they found was the maximum lifespan in these fruit flies was significantly increased, okay? Increased by the overexpression of both SOD and catalase. That gives more credibility to how increasing SOD and catalase can help improve our overall health. So in these experimental models, it actually allowed them to live longer. Leading strong support to the free radical theory of aging. Other work in mammals suggested a possible correlation between SOD and or catalase expression and lifespan. Very recently, this author, Schreiner et al., have significantly extended both median and maximal lifespan in transgenic mice expressing catalase targeted specifically to the mitochondria. Then the last sentence I think is pretty powerful. In view of this work, perhaps the free radical theory of aging should be renamed as the oxidative stress theory of aging. And I think those are really the major takeaways from this study. So again, a really short work, only 3 figures, just a couple of pages long, but an incredibly powerful story, not just in the kind of macro view of this paper, but a lot of that minutia that we can tease out of here just really goes to illustrate how powerful this product is that we have in our hands. And with that, Dondee, I'll hand it back over to you. And if we have any questions or things I can maybe clarify a little bit better, I'd be more than happy to do that.
Thank you so much, Dr. Brian. One of the things I thought was just fascinating, and I'm so glad that you brought that out of the study, was the self-regulating part of it. And with my background in chiropractic, that was one of the main things that we taught patients, is that working with you is going to allow your body, which is constantly striving for this thing called homeostasis, balance, cellular balance in your body, or your body's pursuit of health. What medicine does, and this is, I just thought this was absolutely great. You brought this out of the study. Medicine will take a certain spot in time. And if you say you have high blood pressure, as an example, you can be given medications to lower your blood pressure. But there are also people that have low blood pressure that may have be given a medication to raise their blood pressure. But if you allowed that person's body to self-regulate, then it could figure out exactly what it needs and handle the problem. And so interestingly enough, when there's people that a whole broad spectrum, and that figure 1A, where it was time on Protandim and the age of the people, or the age, and then hey, sorry, I got my dog in the background. Coco. It was pretty amazing how that average line, there was people at both young and old ages that were on the lower side of oxidative stress and some on the high side. It wasn't necessarily correlated directly with age. There's other variables, but it was kind of like, if you're talking about somebody when they're talking, this is kind of to my point about self-regulation. When someone starts taking any of our products, and health is a continuum where you've got optimal health on one end and death on the other, all of us are somewhere on that spectrum in between. Wherever we are, the closer we are to health, or the farther apart away we are from health, or closer to disease or ultimately death, whatever that cause is going to determine where our products are going to meet them. So it meets them where they start, because some people will say, well, I tried it and I felt something that same day. And there's other people that say, well, I tried it and it's been months and I haven't felt anything. It's not, every single person, if you can kind of look at it this way, if we're all driving the same model car, the same make, the same everything, and we're on the same, and we only go 55 miles per hour, but if our goal is to get to New York City, if you start in New Jersey, you're going to get there much quicker than if you started in LA. And so when people just understand an analogy, if you're in LA and you're trying to get to New York City, in other words, you're extremely unwell and you're trying to get to health, it may take a while. Don't think by tomorrow, if you don't feel something, it's not working. But if you're a competitive triathlete, maybe you're already really close. So it's something different. So I love the self-regulation part of this. And to a lot of people, it's amazing because they're wanting a medical outcome when they're looking for, but a medical outcome and a health outcome are very different. A medical outcome is targeting one specific thing, changing it in one specific direction, whereas self-regulation will go wherever the body needs. And when we have products that facilitate and enhance our body's ability to self-regulate, every person's going to have a different experience based on where they are when they took their first Protandim Nrf2. And I love the fact that the study illustrated that. And I hope people truly grasp that concept, because we're all trying to take a similar approach to I took something today, I had a headache, I took some Advil, and my headache was gone in 30 minutes. Everybody wants that really rapid response, but the body doesn't work that way in a health perspective. It only works that way in a medical perspective, and we don't have medical products. And I think people do that. The way you illustrated that, Dr. Brian, was fantastic. Thank you so much for that. And that was, I saw stuff in that study, and I've read that study literally probably 50 times. You pointed out some things and illustrated some things that I glanced over every one of those 50 times that I wish I would have stopped and looked at every little thing. Thank you for bringing that out. That was awesome.
Yeah, and I appreciate that, and that's beautifully put. And maybe to build on your car analogy just a little bit more. I mean, say two cars had to go from LA to New York, but imagine one individual's car. They were really good at maintaining it, changing the oil, doing all that regular maintenance that they needed to do. I mean, what are the odds? Eventually, the individual's car who wasn't maintained is going to start to break down. And really, what we're trying to do is be those mechanics for our own bodies to be proactive in our health and keep that car not just running to New York City, but racing to New York City.
Absolutely. Some of the comments in the chat, Amy and Rachel Hughes ask, where can you get the full copy of the study? Pubmed.gov. Just type in Protandim. And if you scroll, this is the very first study. So chronologically, it will be number 28 if you're looking at it in date of publication. Otherwise, if you look at the title of the study, which again was the induction of human superoxide dismutase and catalase in vivo, just scroll until you find it. But if you type in Protandim in pubmed.gov, you'll see 28 studies come up. It's in there. You can look at the entire study from there.
Hey, Dondee, do you mind why don't we give a quick little tutorial on PubMed, just 2 minutes maybe, just to kind of walk people through what that might look like, just in case you're
be awesome. Yeah, go for that.
Not familiar. Okay, so exactly like Dr. Nettles said, if we come to pubmed.gov, again, this is set up by our National Institutes of Health, and the whole goal is to collate every single scientific and medical publication that's been published related to human health, however directly or indirectly it might be. There's a lot of ways you can search on PubMed. I mean, if we look in here, look, there are currently over 33 million different references that PubMed is collating. But you can search by keyword. You could search by author, for example. If you just type in my name and first initials, you'll see some of my publications here. But if we want to look for a publication, we can just simply type in Protandim. Okay, and we'll start to look at all the different articles that are related to Protandim. An easy way to find the specific article you're looking for is put some of the key, maybe the more unique keywords that might be in the title. So in this case, the induction of human superoxide dismutase. I'd put catalase in there. Catalase. And we'll just see what comes up. Didn't quite get us there. So we can type in a few more words. New approach to therapy. And there it is right there at the top. So the more words you put into the search, the more you can hone down. And then there you go. There's the article. There'll be some quick links up here to get to the actual article. But there can also be, I imagine this this journal is particularly bad about making you pay, but if you get pretty clever in your search on the internet, you'll probably be able to find a copy that you can access.
Great points. And that's one of the things that we can do as well. Like Dr. Dixon was saying in that search, if you just typed in oxidative stress or Nrf2 or other things specifically, you can see how much science dedicates their time studying this thing, because it really comes down to relevance. If we're talking about something that nobody really cares about or nobody studies, then it would be a moot point. But if you just, for example, typed in Nrf2 and hit enter, you'll see there's hundreds of thousands of studies, which means lots and lots of people think that this is something noteworthy enough to study. And when we have a product that contains the very way of triggering that pathway, we have a pretty exclusive approach to handling something that a lot of people pay a lot of attention to, especially scientists, researchers, as it relates to human health. That brings up something, another question that I had, Dr. Brian, and maybe other people have. When you type in Protandim, you'll see we have 28 studies. When this one that you just went through came out, it was literally revolutionary. The ability to lower oxidative stress in a botanical form, which created lots and lots of interest. One of the things now that was great in the beginning and is now, it's still great for us, but can be somewhat problematic. And that is, of those 28 studies, there's a fair amount that deal directly with disease. And as you pointed out, that oxidative stress has been linked to over 200 diseases. We reword that to health challenges, or however we want to say that, to remain compliant. But the connection nonetheless exists. And when people saw that and saw the ABC primetime and saw this publication, I think it opened up a bunch of hypotheses in their mind. What could we do by taking this herbal blend? And they obviously go to pathology, which, if you think about it, it would be a natural progression, because if Nrf2 was so widely studied and we have the number 1 and number 2 killers of human beings being heart disease and cancer, which are very prevalent, you would think somebody somewhere would want to do something good and connect them. And we obviously have heart disease studies. We have cancer studies. They were done on behalf of our product. This is the other thing, third-party initiated, third-party funded. But here's my question in that kind of a historical perspective there, Dr. Brian. What would you say now, and have you noticed a change or a pivot in direction from the company's perspective on allowing our products to be studied on pathology or diseases versus more of an approach to overall human wellness or optimizing human performance?
Well, for right or wrong, you'll either love or hate my answer. But I'm a big fan of being able to publish studies and patents that can actually be utilized. So I think to your original point there, where we do have some of these studies that talk about heart disease or cancer, that gets really tricky to talk about in a compliant manner. So something I've kind of taken upon myself is just to make sure whatever our experimental model might be, I want to make sure that it's something we can talk about. And by experimental model, I've also made the mandate that we're not going to do any animal testing. Like, we want to know our products work in humans, so why not just go to the human condition and test there? So all we're going to be doing in the future, cell culture studies and human studies, but we're also going to set up those experiments so that they can easily be talked about, easily be shared. And I think we're going to see a pretty cool study next week that just was released yesterday evening, centered around Protandim NAD synergizer. And I think you'll start to see how when we use the right words and choose the right experimental models, we can provide materials that you can directly speak to. So that's been our approach. And why a lot of studies do have to go to heart disease or cancer is you do need some sort of compromised population, because again, by definition, it's hard to make somebody who's already healthy healthier. So scientists love to look at extremes to see if we can kind of bridge those gaps in between. It just makes the research a little bit cleaner. But that's a long way to say we're in the business of generating scientific works and patents that are utilizable, if that's a word, utilizable by our field and our distributor base.
Awesome. I have a question, Dr. Brian, and I hate to spring this on you, but it's something that I think about, and I want to know if I'm the only person who thinks about this or has seen this connection. For those of you who know who Dr. Joe McCord is, who obviously who we are at LifeVantage, what our Protandim Nrf2 Synergizer is, you know that Dr. McCord received a very prestigious award called the Elliott Cresson Medal back in 1997. In fact, it was the Elliott Cresson Medal, that was the last year that was issued because the foundation changed. It's now the Benjamin Franklin Foundation, and now the award, which is the same as what the Elliott Cresson Medal used to be, it's now called the Benjamin Franklin Award. But why do I mention this? Because in 2016, Dr. Douglas Wallace received the Benjamin Franklin Award for his study of mitochondrial DNA. And I thought that was interesting because if you've seen the original ABC Primetime, Dr. Douglas Wallace was the guy then at UC Irvine talking about animal studies. He was the guy that said, well, if we've extended the life of mice by 50% in a laboratory, what could that mean to human beings? That guy used to be with Protandim, or used to be with LifeVantage, did studies. He received the modern version of the Elliott Cresson Medal in 2016 for mitochondrial DNA. We have a product, Protandim Nrf2, that deals with mitochondria. A year later, the Nobel Prize in Medicine was given to a doctor out of Japan for his discovery of autophagy. And many people understand autophagy in the keto community or the intermittent fasting community as it relates to sirtuins, as it relates to NAD. So I guess what I'm trying to say is when you have products that deal with things that have been given some of the most prestigious awards in science, maybe, just maybe, we're on the right track. How would you comment on that, Dr. Dixon?
I just absolutely, I couldn't agree with you more. I mean, Joe McCord, it probably won't be Joe McCord. It'll be his graduate mentor at the time, Erwin Friedovich, is probably going to win a Nobel Prize for that discovery of SOD as well, just to layer on top of that. I mean, yeah, that's how important these findings are. Even today, right, you saw that free radical theory of aging was first proposed, 1954 to 1957, depending on how you count. And it's still a major focus of research. And yeah, to that point, just for fun, I went into PubMed and pulled how many articles come up when you look for oxidative stress. It was over 250,000 publications come up. It was like 275,000 articles come up related to oxidative stress. So right there alone, you can see just how important that is. And mitochondria and that free radical theory of aging really have kind of morphed together. And then this thing that's been blown wide open with NAD and our superior approach to increasing those levels in our body, just ripe for promise and discovery in the future.
Yeah. So for those of you who maybe don't understand the significance or the relevance of our science, just know this: for the people that look at science and think it's a big deal, they are looking at what we are working with, the pathways that our products are optimizing, as really, really big deals. So don't have to worry about becoming a mini junior scientist to understand it. Just know that the people who look at relevant areas of study are looking at the very 3 studies that we happen to have products to market for. That is a massive feather in our cap, everybody. So if you are here at LifeVantage and you're part of the LifeVantage family, consider yourself blessed, because no other company and no other group of people has been in the situation that we are in right now at this time ever in recorded history. We are sitting in a very, very unique situation to change modern history and change modern human health. And we have awesome people to work with, like Dr. Dixon. Thank you guys so much for attending, Dr. Dixon. Thank you again for just opening up that massive gray matter of yours and sharing it with us. Thank you for, as always, you're just incredible. We are blessed to have you. Thank you so much for sharing today.
Thank you, Dr. Nettles. And thanks for hosting again. Beautiful job. Thank you.
Thank you, everyone. This will be, as this gets uploaded, eventually it will be onto the Vision Summit YouTube channel. Take a look for it there if you want to send this out. Thank you all for your time. Thank you for your patience and grace in me handling what hopefully I rectified the situation and came around okay. But thank you all for attending. It was awesome. Have a wonderful day. We'll see you guys